Okay, welcome to podcast 5 in our evolution series. We are still looking at evidence for evolution. We have already looked at the biogeography and the fossil record as evidence that evolution, change of species over time on the planet has actually occurred. We are now going to look at comparative anatomy as another block of evidence for evolution. Okay, so please remember again that as we talk about evolution and its evidence, it is not just drawing from one area, but many different areas, and these areas weave all together to make this incredibly impressive body of evidence that evolution has occurred on planet Earth. Okay, now, what is comparative anatomy? Comparative anatomy is simply comparing different body parts in different species and looking for similarities and differences in those body parts. Okay, so if evolution has occurred or has not occurred, there are certain hypotheses we should be able to make, predictions, what we would think we should see. Okay, so if species have actually undergone evolution from a common ancestor, then their anatomy should show that. That should reveal certain fundamentally common structures and common developmental patterns in their body parts. Just like you are more similar to your parents than you are to perhaps a random stranger. But if a species did not evolve from a common ancestor, then there's no reason to expect that the different body parts they have, for example, wings and legs, would share any fundamental structural similarities. They're very different structures used for very different things. Now, what we actually find when we look at comparative anatomy is that species that are grouped in the same taxonomic category, for example, mammals in their group, birds in their group, have lots of anatomical similarities. For example, many of the same bones that make up the forelimbs of humans, like my arm and a cat's foreleg, whale's flippers and bat wings, they have many of the same bones in all those wildly different structures. Okay, of course, those structures have vastly different functions. So it makes not very much sense that the very best way, the ideal way to design a bat's limb for flying and a whale's fin for swimming would be to make those from the same bones. But that is what we see when we look inside the anatomy of a bat wing and a whale fin and a human arm. The same bones are all present. The most likely explanation for seeing that is that all these animals once had a shared common ancestor that had those bones. And since that common ancestor, those bones have become modified for different functions in different groups of organisms over time. Okay, so what we were just talking about is called homologous structures. You need to know this term here. Okay, it comes up again and again. Homologous structures. Homologous structures are similarities in characteristics that result from common ancestry. So these are real clues to evolutionary relationships. These organisms are probably related at some point in the past. Okay, so let's look. From a bat wing to a human forelimb, all these mammalian vertebrate forelimbs, okay, we see the very same bones in each one. All right, the humerus being highlighted here, here, and here, okay. The paired bones, radius and ulna, are all present in all of these animals. Radius and ulna the same. The carpals of the wrist, all present in all of these animals, and perhaps most interestingly, fingers, phalanges, are present, and of course in humans, but in cats, in whales. Why would there be phalanges, fingers, in a whale flipper? It only makes sense that perhaps some ancestor had these digits initially. And of course, this bat's wing is using its highly delicate phalanges to spread out its wing and use it for flight. Homologous structures are structures that have similar developmental patterns. If you look at their embryology, if you look at them during their development, during the embryonic stages of an organism, they have similar parts, like those bones, though they have been modified. They aren't identical. But homologous structures have different functions. They aren't obviously the same structure on the face of it. Okay? They are absolutely evidence of a reasonably close evolutionary relationship. Remember, this is all relative. You know, a cat and a human are closely related if we are comparing a human and cat versus a human and a bird, for example. And they share a reasonably recent common ancestor. Homologous structures are evidence of this, okay? And that's important that you know that. Homologies are evidence of common ancestors, okay? And we see these homologous structures not just in animals, but also in plants. Leaves, for example. Leaves have taken many modified forms that in reality we wouldn't even really recognize them in a leaf, and they function in very different ways, but all these different forms all originated originally from leaves. For example, needles in a pine tree are leaves. They do photosynthesis, but they are modified to reduce surface area and thus reduce water loss. For conifers, pine and family, other members of that group, when the soil is frozen. They don't lose their leaves, they keep their leaves, but they can't get water from the ground, and so they have this reduced surface area, so they won't lose so much water through transpiration. Succulent leaves in more desert or arid type species that store water for the plant. And then, of course, there's the truly desert-dwelling cactus plants whose leaves have become modified into these spines, not only to reduce surface area, but also obviously to defend the plant against animals that are desperate for a water source. And of course, cactus store water in their tissues. Tendrils are modified leaves that are used for climbing so that a plant can get up closer to the sunlight and not be hidden in the shade. And then finally, there are colored leaves that we call that are going to draw in a pollinator to help a plant pollinate, cross-pollinate between different sexes. So all of these structures don't necessarily look identical, but they are all fundamentally leaves. They have just been modified for different purposes. Now, please do not confuse homologous structures with analogous structures. Analogous structures, and you will need to know that term too, are the result of convergent evolution, okay? So it is the separate evolution of structures that end up looking kind of similar, but they aren't really related. Okay, so these structures may have very similar functions, like this insect wing on a dragonfly and this bird wing on this red-tailed hawk. They do the same thing. They enable the organism to fly. They have a somewhat similar external form. They're broad, they're flat, they look like wings. But in analogous structures, the internal structures and the development of the structures is fundamentally different and bears no relationship. There's nothing like a bone in this dragonfly wing, whereas, you know, obviously bird wings have bones. They have bones that are not altogether different from those mammal bones we were just looking at. So these organisms are of different origin. Analogous structures are not evidence of common origin. They are not evidence of common origin, okay? There is no evolutionary relationship, certainly at least not a close evolutionary relationship between dragonflies and hawks. So analogous structures and convergent evolution is solving a similar problem with a similar solution, but it does not indicate evolutionary relationships. Okay? Please don't be fooled by their looks. Analogous structures look like they ought to be related because they're doing the same thing, but they are not. Okay? Now, convergent evolution, which you just mentioned, is how we get those analogous structures. So basically, convergent evolution is when similar environmental pressures are sculpting not related organisms, unrelated organisms, in similar ways. Okay? So let's think about fish. Fish are aquatic vertebrates. They were the original vertebrates. Okay? And fish look like this. They have fins, big fins, small fins, whatever, streamlined bodies, and so on. They are adapted for moving in the water. Dolphins are also aquatic, but they are mammals. Okay? But if you look at them, they also have these smooth, streamlined bodies. They also have fins, and so on. And superficially, they look rather similar to these fish. But of course, in reality, they are not closely related, at least not terribly closely. Yes, they're both vertebrates, but mammals and fish, mammals were originally land animals and have reverted back to the water. Okay, so not terribly closely related. Those fins and tails and sleek bodies are analogous to each other. They are solving a similar problem, but they are not evidence of common origin. Okay. Comparative anatomy also shows us something called vestigial structures. Vestigial structures or vestigial organs are structures that an organism has, but they're kind of useless. They don't have very much importance to the organism. What they seem to be are remnants of structures that once had important function in the organism's ancestors. So, for example, we see in certain whales these vestigial or useless hind limb and pelvic bones that are useless in a water-based swimming animal, but had use in the whale's once ancient land ancestor. Okay? So, vestigial structures are found in modern animals and they have little or no function. Remnants, we already said that. So, how do these, what happens to these vestigial structures? How do they get this way? Well, if an animal is not really using a structure, for example, if a water-based whale ancestor was not really using its hind limbs, then if it gave birth to a baby proto-whale with mutated, badly formed hind limbs, it wouldn't really matter to that baby whale because it wasn't using the hind limbs anyway. So the idea that deleterious, meaning bad, mutations can accumulate in the genes for these non-critical kind of useless structures, and they don't reduce fitness. So these things these structures can kind of be allowed to get useless because bad mutations don't really, the environment doesn't select against them anymore. Examples, snakes and whales. We see in snakes as well as whales those pelvic bone remains. Eyes on blind cavefish. Here's a close relative of certain cavefish. You can see it's highly obviously functional eye, but in cavefish that live in the dark, we see the eye is just gone. And why should they have an eye? They live in a cave where it's dark. They couldn't use it anyway. So those are the eye remnant is vestigial. The human tailbone, you have vestigial structures as well. This structure here, these pitiful 4 vertebrae, serve no function. They aren't supportive. They don't do anything except possibly hurt if you fall on them. So the human tailbone is a classic example of a vestigial structure. Our ancestors once had a highly useful tail. We no longer do. Okay? So vestigial organs are also not only evidence of evolutionary change, but they are evidence of natural selection. Think of it in this way. it would be wasteful to continue to provide nutrients and so on to an organ that really has no major function. Why would you do that? That's a waste of resources. Hind limbs on aquatic animals, why would we support those? Natural selection then would tend to favor individuals who didn't waste their important resources on supporting such organs. And so those organisms survive more, reproduce more, and slowly over time, those organs just get eliminated. Okay, so again, why would whales have pelvis and leg bones if they had always been sea creatures? So comparative anatomy, homologous structures, analogous structures, and vestigial organs. Homologous structures and vestigial organs are evidence of common ancestry. Analogous structures are not. Okay, that's it. Thanks for watching. See you in the next podcast.